So picture this. You're walking barefoot in your house. Maybe you're half asleep, and suddenly you step squarely on a stray thumbtack.
Oh yeah, the absolute worst way to wake up.
It really is. And before you even have time to yell, or before your brain has even, consciously registered the feeling of pain, your foot has already violently jerked back into the air.
Right, it's that instantaneous recoil.
Exactly. We've all experienced it, but according to this really fascinating 2026 paper from the journal Neuron, we didn't actually know which specific nerve wires pulled that off until right now.
Which is, honestly, it sounds counterintuitive, right? Given how fundamental pain is to our basic survival.
You'd think we'd have mapped that out decades ago.
You really would. I mean, we've always known the recoil happens, that involuntary, lightning-fast protective move, but tracing the exact biological hardware driving that specific response has been a massive hurdle for researchers.
There's a mess of wiring in there.
Exactly. The field knew there was an emergency alarm system, of course, but finding the exact physical cables in just the sea of nervous tissue was incredibly elusive.
Well, that perfectly sets up our mission for today. We are taking a deep dive into this new research to uncover the actual physical wiring of acute mechanical pain.
It's such a great paper to dig into.
It really is, because the researchers finally identified the specific fast-conducting nerve fibers that act as the body's dedicated emergency responders. So, okay, let's unpack this, because historically, isolating these specific wires in a lab was a serious problem.
It was a huge bottleneck.
Right. Because you can't exactly hand a mouse a pain scale clipboard and ask it to rate a pinch from 1 to 10.
No, you really can't. And that is the core bottleneck of preclinical pain research. Without verbal feedback, scientists previously just had to guess which sensory neurons were the true nociceptors, the actual pain receptor.
So how were they guessing before this?
Well, they would look at broad molecular markers or honestly just measure the physical thickness of the cells. And by doing that, the field mostly focused its attention on a massive group of neurons called C fibers.
Okay, the C fibers.
Yeah, these are tiny unmyelinated nerve fibers. And unmyelinated just means they lack the fatty insulating sheath that normally speeds up electrical signals in the body.
So they're slow.
Very slow, making them very slow conductors. But for decades, the assumption was just that these slow C fibers were doing the heavy lifting for pain.
So they're essentially looking at a giant bundle of different colored cables and just guessing that the alarm system was the thinnest wire in the bunch.
That's a perfect analogy, yeah.
But this new study completely bypasses that whole guessing game by using optogenetics. And for those of you listening who haven't encountered it, optogenetics is this wild technology where scientists genetically modify specific neurons to react to light.
It's basically science fiction brought to life.
It really is. But I want to understand the actual mechanics of what they did here, because they engineered 10 distinct types of sensory neurons in the mouse paws to express something called reacher.
Right, reach.
So how does shining a light actually force a nerve to fire?
Well, it comes down to cellular engineering. So reacher is a specialized light-gated ion channel. The researchers genetically coded these specific neurons so that they would physically build these tiny channels into their cell walls.
Okay, so they're inserting basically tiny doors into the cell.
Exactly, tiny locked doors. Now, normally a nerve fires when mechanical pressure or heat forces its regular ion channels to open, which lets positively charged ions flood into the cell.
Right, just natural physical force.
Yeah, but with reacher, that pore is physically pulled open by the energy of blue light. So when the laser hits the tissue, the channel snaps open, sodium rushes in, and an electrical spike in action potential shoots right up the nerve.
Oh, wow. So they can trigger the nerve with zero physical touch, zero heat, and zero actual tissue damage. They just flash a laser.
It's just a flash of light.
And the time frame here is what really catches my attention. They used a simple 5-millisecond pulse of blue light. I mean, that is an incredibly short window.
It's just enough time to manufacture a single electrical spike.
Just one spike.
Just one. And that single spike led to the definitive aha moment in the lab. Because when they shined that laser on the famous slow-moving C fibers, the results were entirely underwhelming.
Really? Like nothing happened?
Often the mouse didn't react at all. And if it did react, it was this sluggish withdrawal, pulling the paw away after a long delay of around 200 to 400 milliseconds.
Which definitely wasn't the violent, instantaneous jerk you experience when stepping on a tack.
No, not at all. It was way too slow.
Which pushed them to test a very different set of wires, right?
Exactly. They isolated 2 specific populations of fast-conducting, heavily myelinated nerve fibers known as Aδ-HTMRs.
Okay, in the paper they track these using 2 specific genetic markers. I have them here. Is MR2 and BIMBRO1B.
Yep, those are the two.
So what happened when the 5-millisecond laser pulse hit those specific cells?
The reaction was explosive. The latency, the actual reaction time from the light turning onto the muscle physically moving was just 20 milliseconds.
That is absurdly fast.
It's a blink of an eye. The mice jump, they shook their paws, they aggressively guarded the limb. They even learned to avoid the side of the testing chamber where the laser was positioned.
Oh, so they were actually afraid of that area.
Exactly. That behavioral avoidance proves this wasn't just some unconscious muscle twitch. It possessed a highly aversive emotional quality. They absolutely hated the sensation.
So if these alarms require such an explosive reaction, their natural triggers in the real world must be pretty extreme, right?
Oh, yeah, they have to be.
Because, I mean, we interact with our environment constantly. We put on socks, we walk on carpet, we type on a keyboard, and we don't feel pain.
Right, it's mostly just background noise.
So if we take away the high-tech laser, what kind of physiological force does it actually take to get these Aδ-HTMRs to fire?
Well, they tested that exact question by measuring the physiological thresholds of these neurons. And it turns out they are intensely exclusive.
What do you mean exclusive?
I mean, they remain completely silent during gentle brushing or normal tactile exploration. To force these specific ion channels to open naturally without the laser, the researchers had to use stimuli that essentially threaten the structural integrity of the tissue.
Like actual damage?
Yeah, like they used stiff von Frey filaments, which are these specially calibrated nylon hairs designed to bend only at a very specific high-pressure force. They used heavy pokes with closed forceps. They used strong mechanical pinches.
So they basically sit completely dormant, just ignoring all the background noise of daily life until they encounter a mechanical force strong enough to potentially break the skin.
Exactly. It's a true emergency brake.
That makes perfect sense for an alarm system.
Yeah.
And that high threshold requirement really makes me wonder about their physical placement.
Yeah.
Where exactly is the body laying these specific tripwires to optimize for danger?
The researchers found them branching out expansively in glabrous skin, which is the hairless skin on the palms of your hands and the soles of your feet.
Okay, the touch points, basically.
Right, the surfaces that interact with the world the most. But the truly vital detail is their unique architecture within that tissue. They branch out and penetrate all the way up into the epidermis, the very outermost layer of the skin.
Right at the surface.
Right at the surface.
Mm-hmm.
And they don't just terminate as single isolated endpoints. They form this highly dense overlapping meshwork. In any given tiny spot on the hairless skin of the paw, there are 3 to 10 of these Aδ-HTMR nerve endings overlapping and tangling with each other.
Wait, hold on. If I have 10 alarms all tangled up and overlapping in 1 single millimeter of my foot, doesn't that create a massive biological bottleneck?
How so?
Well, why would evolution waste so much energy building 10 redundant wires when one would do the job? It seems like having them all tangled would make it incredibly hard for the brain to pinpoint the exact localized source of the pain.
Oh, well, you've zeroed in on a fundamental biological trade-off between spatial resolution and signal reliability.
Okay, break that down for me.
Consider the nerves responsible for fine discriminative touch, the ones that allow you to reach into your pocket and feel the difference between the smooth edge of a nickel and the ridges of a quarter.
Right, you need extreme precision for that.
Exactly. Those fine touch nerves have very tight, strictly non-overlapping territories. Your brain needs that high-resolution mapping to pinpoint exact textures.
So when you step on a shard of glass,
yeah,
you don't need to know the intricate texture of the glass.
right, or the exact nanometer of skin it's piercing. You just need to get your foot off the floor right away.
Oh, so you sacrifice spatial accuracy for absolute certainty?
You do. Having 3 to 10 overlapping alarms guarantees that no matter what angle the sharp object hits, or if one tiny nerve ending happens to be damaged, the distress signal will definitively make it to the spinal cord. In an emergency, the nervous system completely prioritizes redundancy over resolution.
That is brilliant.
Yeah?
Okay, but turning them on with a laser proves they can trigger the alarm. But how do we know they are the only alarm? Like, if you take these specific fibers offline, does a backup system kick in, or does the whole emergency reflex just collapse?
To test that, the researchers conducted an ablation experiment, meaning they selectively destroyed these cells to see what the mice would do without them.
Okay, and how do you selectively destroy just one type of nerve.
The methodology here is brilliant. They use diphtheria toxin, or DTX. Now, mouse cells naturally do not have the receptor required for diphtheria toxin to bind to them.
Okay, so it wouldn't hurt a normal mouse.
Right, it would just wash right through. So the scientists genetically engineered the mice so that only the Aδ-HTMR cells, the ones expressing those FMR2 and BMPRE1B genes, would grow the diphtheria toxin receptor.
Oh, wow. That is an incredibly elegant genetic trick.
It really is.
So they inject the toxin systemically. It washes harmlessly over the entire brain and body. But the moment it bumps into one of these specific high-threshold pain wires, it docks with the engineered receptor and destroys the cell.
Exactly, leaving the rest of the entire sensory nervous system entirely intact.
So what happened when they snipped the wires of this specific alarm system?
The mice lost about 60% of their reflexive response to sharp pinpricks. The behavioral change was stark.
I was looking at the results of that ablation setup, and what surprised me wasn't just that they stopped reacting to pinpricks, but how incredibly isolated the effect was.
Right, they didn't just go completely numb.
No, the researchers noted the mice could still sense moderate hot and cold temperatures, and they still actively avoided rough sandpaper, which proves this isn't some master pain switch for the whole body.
No, it's a highly specialized, dedicated subroutine strictly for acute mechanical force.
But living without that specialized subroutine came with a severe cost, right?
A very severe cost. Without these fast mechanical alarms, the mice began developing facial and bodily wounds simply from going about their normal everyday activities in their cages.
Just from normal life.
Yeah, they were just accumulating all these microtraumas.
Because they lacked that instant feedback loop. Like if you can't feel the sharp edge of a food pellet against your lip or a rough edge on a water bottle, you don't micro-adjust your posture or your bite.
You just keep pushing until the tissue literally breaks.
It's a perfect biological mirror to human patients with genetic pain loss disorders. They frequently suffer severe joint damage and skin wounds because they lack this exact guardian mechanism. We view pain as an antagonist, but it is fundamentally the architect of our physical boundaries.
The physical boundaries of the skin, yes. But the deep dive into the Sork material revealed something even more expansive, actually.
Oh, really?
Yeah, these specific Aδ-HTMR fibers aren't just a surface-level defense system. They densely innervate several unexpected internal organs as well.
I was wondering about that. Because a thumbtack is external, but internal pain, like when something inside your body stretches or tears, is a completely different sensation.
It feels different, but structurally it relies on the exact same hardware. The researchers found dense innervation in the knee joint, specifically within the meniscus and the infrapatellar fat pad.
Oh, man, which instantly demystifies why a sharp mechanical twist of the knee during a run can drop you to the floor in agony before you even register the popping sound.
Exactly, it's those same fast fibers. They also found these overlapping nerve meshes densely packed in the urinary tract, specifically the renal pelvis of the kidney and the ureters.
Wait, the ureters are the tiny tubes connecting the kidneys to the bladder, right?
Yes, they are.
So we are talking about the mechanism of kidney stones.
We absolutely are. Think about it. A kidney stone is essentially a jagged, solid mass trying to force its way down a highly restricted muscular tube.
Which sounds horrific.
It is. The stone creates extreme mechanical distension. It severely stretches the tissue and applies immense localized pressure.
And that pressure violently physically deforms those overlapping Aδ-HTMR nerve endings.
Exactly. Knowing these specific high-threshold fibers are lining that tube gives you a visceral understanding of why passing a stone is considered one of the most agonizing mechanical pains a human can endure.
Yeah, that makes total sense now. And the innervation didn't stop in the lower body, did it?
You know, they trace these same SMR2 and BMPRE1B fibers into the trigeminal ganglia, which is the nerve cluster serving the head and face.
Okay, into the head.
Yeah, they found them wrapping into the cranial meninges, the membrane surrounding the brain, and diving deep into the tooth pulp.
Wow, so toothaches and migraines too.
Identifying these fibers across such vastly different internal landscapes points to a truly universal biological mechanism. Whether the intense mechanical pressure is coming from a thumbtack, piercing the heel, a jagged stone, stretching the ureter, or inflamed tissue compressing a nerve inside a tooth, your body relies on the exact same specialized Aδ-HTMR hardware to sound the alarm.
The universality of it is just fascinating, but it actually brings me to a central mystery about the circuitry itself that I can't quite reconcile.
What's that?
We talked about a 20 millisecond reaction time earlier. How does a signal get from the bottom of your foot, travel all the way up the spinal cord to the brain, get consciously processed, and then travel all the way back down the spinal cord to the leg muscles to orchestrate a physical withdrawal all in 20 milliseconds.
It's incredibly fast.
It's too fast. The physics of nerve conduction speeds just don't add up for a round trip of that distance.
Well, you're right. The math doesn't add up because the signal never makes the round trip.
Well, what do you mean?
To prove this, the researchers performed a dramatic but completely necessary experiment. They did a spinal transection. They completely severed the spinal cord of the mice at the T9 vertebra right in the middle of the back.
Meaning they physically cut all communication between the hind legs and the brain. The brain is entirely isolated from the lower half of the body.
Exactly. And yet, when they flashed the optogenetic laser on the toe of the transected mouse, the ultra-fast paw withdrawal still happened with the exact same speed and intensity.
Oh, wow. So it's like edge computing in modern tech networks.
That's a great way to think about it.
Yeah, instead of sending all the raw data back to the central cloud server, your brain, which takes precious milliseconds, the spinal cord just acts as a local decentralized router.
It receives the alarm, processes the danger, and makes the executive decision to fire the muscles instantly, completely bypassing the brain's processing lag.
That is amazing.
That local reflex circuit is what allows for the 20-millisecond reaction. The signal travels from the toe to the base of the spinal cord, synapses directly with the motor neurons, and shoots right back down.
Okay, but however, as you know from stepping on a tack, you do eventually feel the pain. Your foot jerks up, and a fraction of a second later, the conscious suffering hits you.
Oh yeah, the brain is undeniably involved in the emotional aftermath.
So how does the signal split? Because these fibers have to be talking to the brain somehow to create that memory of the pain.
This is where the specific anatomy of these Aδ-HTMRs inside the spinal cord becomes truly unique. Sensory nerves enter the spinal cord at the dorsal horn. Most of the gentle touch nerves terminate in the very superficial outer layers.
Right near the surface.
But these fast pain fibers dive much deeper. They bypass the surface and plunge into the deep dorsal horn where they form these intricate wraparound structures that the researchers literally describe as nests.
Like a bird's nest woven out of nerve axons.
Visually, yes, exactly. The axons of the pain fibers wrap completely around the cell bodies of their specific target neurons in the spinal cord, and they form a monosynaptic connection.
Meaning there is only one single synapse, just one gap to cross.
Okay, there are no middleman interneurons slowing the message down. Furthermore, the cells they are wrapping around aren't just random neurons, they are specialized antenna cells, the projection neurons of the anterolateral tract.
Okay, the anterolateral tract being the superhighway that shoots signals straight up the spinal cord into the brain.
Specifically, they shoot straight up to the parabrachial nucleus in the brainstem.
And what does that part of the brain do?
The parabrachial nucleus is a major hub for processing the emotional, highly aversive qualities of pain, the fear, the suffering, the intense desire to escape.
So the anatomy perfectly matches the function. The nest structure isn't just tapping the antenna cell on the shoulder to casually pass a message along. The Aδ-HTMR is putting the projection neuron in a massive bear hug.
Oh, massive bear hug, yeah.
It guarantees absolute fidelity of the signal, ensuring the message violently shakes the brain awake and says we are taking structural damage, generate an intense feeling of suffering so we never do this again.
It is a brilliant dual action system. You have the local spinal router handling the immediate physical withdrawal for survival, while the monosynaptic bear hug ensures the emotional trauma is registered by the brain for long-term learning and avoidance.
This entire biological mechanism is just staggering when you lay it out from start to finish. Just to recap the journey for everyone listening, we've tracked the body's true fast pain wires, the Aδ-HTMRs.
Right.
We've seen how they form overlapping redundant safety nets in our hairless skin to guarantee the signal gets through, and how those same networks line our internal organs like the ureters and tooth pulp.
And then the spinal cord.
Exactly. We track them diving deep into the spinal cord to form those monosynaptic nests, simultaneously triggering our instant edge computing reflexes and our conscious emotional suffering. We finally possess a definitive genetic map of the exact cables responsible for acute mechanical pain.
And possessing that genetic map leads to the most provocative implication of this entire study.
Where does this lead us?
Well, for decades, treating severe pain has largely meant relying on systemic drugs like opioids, which blanket the entire nervous system. They alter brain chemistry, risk severe addiction, and cause systemic side effects.
Right. It's a blunt instrument.
Very blunt. But now that we have genetic access to these two highly specific fast-acting pain fibers, SMR2 and BAMWAR-B,
We can target the exact wire causing the problem.
imagine a future where we can design targeted analgesics that only interact with those specific ion channels. We could theoretically quiet the exact overlapping nerve meshes in a ureter during a kidney stone attack, or silence the specific trigeminal branches during a severe migraine. We could shut off excruciating mechanical pain directly at the source without numbing a patient's ability to feel a gentle touch, without impairing their temperature sensing, and entirely bypassing the central nervous system risks of traditional painkillers.
That is an incredible thought to leave you with. If medical science can target the exact fiber optic cable, we won't have to shut down the entire city's power grid just to fix a single blaring alarm.
It really changes everything.
It does. Thank you so much for joining us on this deep dive into the source material. The next time you experience that split-second recoil from a sharp edge, hopefully you walk away with a profound appreciation for the dual-action machinery operating beneath your skin. Stay insanely curious, everyone.